Hi class, this is your instructor, Skylar Huff, and we have now made it to what is called Chapter 12. So in this chapter, you're going to learn how genes work. So we'll go from gene to protein, and we'll get into the process of regulation of gene expression. So did you ever wonder what it would be like for you if you could change just one thing about yourself? Some of us desperately want to be healthier. Maybe you want to be taller. Maybe you want to be shorter. Maybe you want to be taller, shorter, stronger. Maybe you want to be thinner. Nonetheless, others would like to be smarter, stronger, more slender. Well, presto, with a wave of almost magic technology, laboratory scientists are moving a long way toward granting our wish. Let us begin. The discovery that genes are made of DNA, discussed last chapter, left unanswered questions about how the information in DNA is used. How does a string of nucleotides in a spiral molecule determine if you have red hair? We now know that the information in DNA is arrayed in little blocks, like entries in a dictionary, and each block is a gene that specifically specifies a sequence of amino acids for a polypeptide. These polypeptides form the proteins that determine what a particular cell will be like. All organisms form, from the simplest bacteria to ourselves, use the same base mechanism of reading and expression of genes, so fundamental to life as we know it, as it is often referred to as the central dogma. Information passes from the genes in DNA to an RNA copy of the gene, and the RNA copy directs the sequence of assembly of a chain of amino acids. Said briefly, DNA to RNA to protein. The central dogma. DNA, RNA, protein. A cell uses 4 kinds of RNA in the synthesis of proteins: messenger RNA, or mRNA, microRNA, ribosomal RNA, or rRNA, and then transfer RNA, or tRNA. These types of RNA are described further. The first step of the central dogma is the transfer of information from DNA to RNA, which occurs when an mRNA copy of the gene is produced. Because the DNA sequence in the gene is transcribed into RNA sequence, this stage is called transcription. Transcription is initiated when an enzyme, RNA polymerase, binds to a special nucleotide sequence called a promoter, located at the beginning of a gene. Starting there, the RNA polymerase moves along the strand into the gene. As it encounters each DNA nucleotide, it adds the corresponding complementary RNA nucleotide to a growing mRNA strand. This chain is a complementary transcript of the gene from which it was copied. The second step of the central dogma is the transfer of information from RNA to protein. This is what occurs when the information contained in mRNA transcript is used to direct the sequence of amino acids during the synthesis of polypeptides by ribosomes. This process is called translation because the nucleotide sequence of the mRNA transcript is translated into an amino acid sequence in a polypeptide. Translation begins when an rRNA molecule within the ribosome recognizes and binds to the start sequence of the mRNA. The ribosome then moves along the mRNA molecule, three nucleotides at a time, called codons, and each group of the 3 nucleotides referred is a code word that specifies which amino acid will be added to the growing polypeptide chain and is recognized by a specific tRNA molecule. MicroRNA molecules help the cell fine-tune gene using the influencing transcripts translated. The use of information in DNA to direct the production of a particular polypeptide is called gene expression. Just as an architect protects building plans from loss or damage by keeping them safe in a central place and assuming only blueprint copies to on-site workers, and issuing only blueprint copies to on-site workers, your cells protect their DNA instructions by keeping them safe within the central DNA storage area, the nucleus. The DNA never leaves the nucleus. Instead, The process of transcription creates the blueprint copies of particular genes that are sent out into the cell to direct assembly of proteins. These working copies of genes are made of ribonucleic acid, or RNA, rather than DNA. The RNA copy of a gene used in the cell to produce a polypeptide is called messenger RNA, or mRNA. It is the messenger that conveys information from the nucleus to the cytoplasm. The copying process that makes the mRNA is called transcription. Just as monks in monasteries used to make copies of manuscripts by faithfully transcribing each letter, so enzymes within the nucleotide of your cells make mRNA copies of your genes by faithfully complementing each molecule. In your cells, the transcriber is a large and very sophisticated protein called RNA polymerase. It binds to one strand of DNA of a double helix at the promoter site, then moves along the DNA strand like a train engine on the track. Although DNA is double-stranded, the two strands have complementary rather than identical sequences. So RNA polymerase is able to bind to only one of the two strands, the one with the promoter sequence it recognizes. As RNA polymerase goes along the DNA strand, it is copying. It pairs each nucleotide with its complementary RNA version. So G with C, A with U. Recall that RNA uses the nucleotide uracil in place of thymine. The mRNA chain is built in the 5' to 3' direction. The essential lesson of genetics is that information determining hereditary traits is encoded in information written in the chromosomes in blocks called genes. To read a gene, a cell must translate the information encoded in DNA into the language of proteins. That is, it must convert the order of the gene's nucleotides into the order of amino acids in a polypeptide, a process called translation. The rules that govern this translation are called the genetic code. The mRNA nucleotides sequence is read by a ribosome in the 3 nucleotide units called codons. Biologists worked out which codon correspond to which amino acids by trial and error experiments carried out in test tubes. In these experiments, investigators used artificial mRNAs to determine the sequence of polypeptides in the tube, then looked at the sequence of amino acids in the newly formed polypeptides. An mRNA that was a string of UUUUU, for example, produced a polypeptide that was a string of phenylalanine amino acids, telling investigators the codon UUU corresponded to the amino acid PHE or phenylalanine. Most amino acids are specified by more than one codon. The entire genetic code dictionary is written for you in figure 12.5. The genetic code is universal. I repeat, the genetic code is universal, the same in practically all organisms. GUC codes for valine in bacteria. Yes, I said bacteria, fruit flies, eagles, and your own cells. The only exception biologists have ever found to this rule is how cell organelles that contain DNA, mitochondria and chloroplasts, and few microscopic protists read the stop codons. In every instance, the same genetic code is employed by all living organisms. The final result of the transcription process is a production of mRNA, a copy of a gene. Like a photocopy, the mRNA can be used without damage or wear and tear on the original. After transcription of a gene is finished, the mRNA passes out of the nucleus into the cytoplasm through nuclear pores of that nuclear envelope. There, translation of the genetic message occurs. In translation, organelles called ribosomes use mRNA produced by transcription to direct the synthesis of a polypeptide, following the genetic code. Ribosomes are the polypeptide-making factories of the cell. Ribosomes are very complex, containing over 50 different proteins and 3 chains of ribosomal RNA, or rRNA, of 3,000 nucleotides. It had been traditionally assumed that the proteins in a ribosome act as enzymes to catalyze the assembly process, with the RNA acting as a scaffold to position the proteins. Powerful atomic resolution X-ray diffraction studies have unexpectedly revealed that the many proteins of a ribosome are scattered over its surface like decorations on a Christmas tree. The role of these proteins seems to stabilize the many bends and twists of the RNA chains. Importantly, there are no proteins inside the ribosome where the chemistry of the protein synthesis takes place, just twists of RNA. It's the ribosome's RNA, not its proteins, that catalyzes the synthesis of proteins. Ribosomes are composed of two parts, or subunits, one nested into the other, like the fist in the palm of your hand. The fist is the smaller of the two subunits, the pink structure that you will see on figure 12.6, and it's the RNA. Its RNA has the short nucleotide sequence exposed on the surface of the subunit. This exposed sequence is identical sequence called the leader region that occurs at the beginning of all genes, because this mRNA molecule binds to the exposed rRNA of the small subunit like a fly sticking to fly paper. Directly adjacent to that exposed rRNA sequence are the three small packets or dents assigned to A, P, and E sites. And those are in the surface of the ribosome as shown there. These sites have just the right shape to bind yet another RNA molecule, transfer RNA or tRNA. It is the tRNA molecules that bring amino acids to the ribosome, used in making the proteins. tRNA molecules are chains about 80 nucleotides long. The string of nucleotides folds back on itself, forming a three-loop structure as shown in figure 12.7a. The three-loop structure folds into further compact with three nucleotide sequence at the bottom, and then an amino acid attached at the attachment site at the top, or the A site. The three nucleotide sequence tRNA, called the anticodon, is very important. It is the complementary sequence to one of the 64 codons of the genetic code. Special enzymes called activating enzymes match amino acids in the cytoplasm with their proper tRNAs. The anticodon determines which amino acid will attach to a particular tRNA, because the first dent in the ribosome is called the A site for attachment. The attachment site for the amino acid-bearing tRNAs will bind. It is directly adjacent to where the mRNA binds to the RNA. Three nucleotides of the mRNA are positioned directly facing the anticodon of the tRNA. Like the address on a letter, the anticodon ensures that the amino acid is delivered to its correct address on the mRNA where the ribosome is assembling the polypeptide. Now let's get to making the polypeptide, y'all. Once mRNA molecule is bound to a small ribosomal subunit, the other larger subunit binds as well, forming the complete ribosome. So if you look at the essential biological process, you all will see the ribosome then begins the process of translation. The mRNA begins to thread through the ribosome like a string passing through the hole of a donut, moving from left to right. The mRNA passes through short spurts, through three nucleotides at a time, or one codon at a time, at each burst of movement. A new three nucleotide codon on the mRNA is positioned opposite the site of the ribosome that the tRNA binds to. As each tRNA brings an amino acid in to each new codon, the old tRNA is paired with the previous codon, is passed along to the adjacent site where the peptide bond forms between the incoming amino acid and the growing polypeptide chain. The tRNA eventually exits the amino acid it carried as it is attached to the end of the growing amino acid chain. So the ribosome proceeds. Down the mRNA, one tRNA after another is selected to match the sequence of mRNA codons, and the growing polypeptide chain, extending out from the ribosome, exits. So translation will continue. I repeat, translation, it will continue until a stop codon is encountered, which signals the end of a polypeptide. the ribosome complex falls apart, and the newly made polypeptide is released into the cell. Now let's get further into gene expression. The central dogma discussed in section 1 of this chapter, being 12.1, is the same in all organisms. So figure 12.9, as it is shown on page 215, presents an overview of the components needed for the key process of DNA replication, transcription, and translation, and the products that are formed in each. In general, the components are the same. The processes are the same, and the products are the same whether prokaryotes or eukaryotes. However, there are some differences in gene expression between the two types of cells. Introns and exons. In prokaryotes, there's a difference. So a gene is an uninterrupted stretch of DNA nucleotides whose transcript is read 3 nucleotides at a time to make a chain of amino acids. And when I say 3 nucleotides at a time, again, I am here referring to codon. A codon is 3 nucleotides at a time. In eukaryotes, by contrast, genes are fragmented. In these more complex genes, the DNA nucleotide sequences encoding an amino acid sequence of a polypeptide, they're called exons. So I repeat, the actual coding regions in eukaryotes are referred to as being exons. It is the exon that actually codes for the protein. So they're interrupted by frequently extraneous nucleotides or extra stuff called introns, or spacers as I call them in those lectures I give face-to-face. In the segment of DNA illustrated in figure 12.10, the exons are the blue areas and the introns are the orange areas. I'm showing the figure for you now. Imagine looking at an interstate highway from satellite. Scattered randomly along the thread of concrete would be cars, some moving in clusters, others individually. Most of the road would be bare. This is what a eukaryotic gene is like. Scattered exons embedded within much longer sequences of introns. In the typical human gene, only 5% of the DNA is devoted to exons that encode a polypeptide, while 95% is devoted to non-coding regions called introns. Processing the primary transcript. So when eukaryotic cell transcribes a gene, it first produces a primary RNA transcript of the entire gene, shown in figure 12.10 as I show you here now. It's in the middle of the screen. So the exons are in green and the introns are in orange. Enzymes add modifications such as that 5' cap and that 3' poly A tail. They protect that RNA transcript from degradation because there are enzymes within the cytoplasm. The primary transcript is then processed. Enzyme RNA complexes excise or cut out the introns and join together the exons to form shorter, mature mRNA transcript that is actually translated into the amino acid chain. This is also known as the secondary mRNA. Alternative splicing. If the introns are not translated into proteins, why have them? It appears that many human exons are functional modules that can be spliced together in more than one way. One exon might encode a straight stretch of a protein, another may encode a curve, or yet another flat space. Like mixing Tinkertoys parts, you can construct quite different assemblies by employing the same exons in different combinations or orders. With this process is called alternative splicing. 20,000 to 25,000 genes of the human genome seem to encode as many as 120,000 different messenger mRNAs. Prokaryotic cells lack a nucleus, so a gene can be translated as it's being transcribed. However, in eukaryotic cells, the nuclear membrane separates the process of transcription and translation, so making protein synthesis much more complicated. And if you look at figure 12.12, it walks you through the entire process, and that is shown for you on page 217. Transcription, step 1, RNA processing, step 2, occur within the nucleus. In other words, transcription and the processing of RNA occur within the nucleus, whereas in step 3, the mRNA travels into the cytoplasm where it binds to the ribosome. So in step 4, the tRNAs, they bind to the appropriate amino acids that correspond to their anticodons. So in steps 5 and 6, as you all can now see here, shown to you all kind of that top left corner, maybe about 11 o'clock, and right there in the center, the tRNA brings the amino acids to the ribosomes, and the mRNA is translated into a polypeptide. Being able to translate a gene into polypeptide is only one part of gene expression. Every cell must be able to regulate when a particular gene is used. Imagine if every instrument in a symphony played at full volume all the time. All the horns blowing full blast, each drum beating as fast and as loud as it could. No symphony plays that way, because music is more than noise. It is the controlled expression of sound. In the same way, growth and development are due to the controlled expression of genes, brought into play at the proper moment to achieve precise, delicate effects. Control of gene expression is accomplished very differently in prokaryotes than in the cells of complex multicellular organisms. Prokaryotes have been shaped by evolution to grow and divide as rapidly as possible, enabling them to exploit transient resources. Proteins in prokaryotes turn over rapidly. This allows them to respond quickly to changes in their external environment by changing patterns of gene expression. In prokaryotes, the primary function of gene control is to adjust the cell's activity to its immediate environment. Changes in gene expression alter which enzymes are present in the cell in response to the quantity and the type of cell available and the amount of oxygen present. Almost all of these changes are fully reversible, allowing the cell to adjust its enzyme levels up or down as the environment changes. Prokaryotes control the process of their genes largely by saying when individual genes are to be transcribed. At the beginning of each gene are special regulatory sites that act as points of control. Regulatory proteins within the cell bind to these sites, turning the transcription of the gene off or on. For a gene to be transcribed, the RNA polymerase has to bind to a promoter, a specific sequence of nucleotides on the DNA that signals the beginning of a gene. In prokaryotes, the expression of a gene is regulated by controlling the access of the RNA polymerase to the gene's promoter. A gene is turned off by the binding of a repressor protein to the DNA, which blocks the access of the promoter. Genes are then turned on by binding of an activator, which is a protein that makes a promoter more accessible to the RNA polymerase. Most prokaryote genes are negatively controlled. They are turned off except when needed. In these genes, the regulatory site is located between the place where RNA polymerase binds to the DNA, that's the promoter site, and the beginning edge of the gene. When the regulatory protein called the repressor is bound to its regulatory site, called the operator, in its presence blocks the movement of the polymerase toward the gene. So imagine if you went to sit down to eat dinner and someone was already sitting in your chair. You could not begin your meal until the person was removed from your chair. In the same way, the polymerase cannot be transcribing the gene until the repressor protein is removed. To turn on a gene whose transcription is being blocked by a repressor, cells use special signal molecules. The binding of the signal molecules to the repressor causes it to contort into a shape that doesn't fit the DNA. The repressor falls off, removing the barrier to transcription. A specific example of how the repressor protein works is the set of genes called the lac operon in the bacterium Escherichia coli. An operon is a segment of DNA containing a cluster of genes that are transcribed as a unit. So let's get to the lac operon here. It's shown on page 219, figure 12.13. So it consists of both protein encoding genes and associated regulatory elements, the operator, the promoter, and other binding sites. When E. coli encounters sugar, lactose, a form of sugar, allolactose binds to the repressor protein and induces a twist in the shape that causes the repressor to fall from the DNA. RNA polymerase is blocked and starts to transcribe the genes needed to break down the lactose to get energy. That is the lac operon. In multicellular organisms with relatively constant internal environments, the primary function of gene control in a cell is to participate in the regulating the body as a whole. Some of these changes in gene expression compensate for the changes in the physiological condition of the body. Others mediate the decisions that ultimately produce the body, ensuring that the right genes are expressed in the right cells at the right time during development. The first hurdle faced by RNA polymerase in transcribing in the eukaryotic gene is gaining access to it. Back in Chapter 8, we described eukaryotic chromosomes that consist of chromatin, a complex of DNA protein. In chromatin, the DNA is packaged with histone proteins into nucleosomes. During mitosis, nucleosomes wind, twist, and coil into chromosomes. But even during interphase, when transcription is occurring, some sections of DNA remain as part of the nucleosomes. These nucleosomes seem to block the binding of RNA polymerase and other proteins called transcription factors to the promoter site. Histones can be chemically modified as a result in greater condensation of the chromatin, making it even less accessible. Another chemical modification of the DNA, adding a methyl group to the cytosine nucleotides. So this blocks accidental transcription of turned off genes. So DNA methylation ensures that once a gene is turned off, it stays off. Eukaryotic transcription requires not only RNA polymerase molecule, but also a variety of other proteins called transcription factors that interact with polymerase to form an initiation complex. Transcription factors are necessary for the assembly of the initiation complex and the recruitment of RNA polymerase to a promoter. There are up to 1,600 different transcription factors known to act in the human genome. Different cells in the body tissues often employ different transcription factors, and one cell may activate different transcription factors at different times. Although prokaryotic gene control sites are positioned immediately upstream of the coding region, eukaryotes can have regulatory sites located far from the gene. And again, far from the gene. Eukaryotic activators can bind at distant sites called enhancers, and DNA bending to form a loop brings the activator in contact with the RNA polymerase or initiation complex. Complex translational control is only one aspect of gene regulation in eukaryotes. Other processes associated with translation, such as gene silencing, protein synthesis, and post-translational modification, are described in the Essential Biological Process 12A, and they provide the cell the ability to produce finely graded responses to environmental and developmental signals. And you can see that shown to you on page 222. As will be discussed in detail in Chapter 13, the bulk of eukaryotic genome is not translated into proteins. This finding was puzzling at first, but began to make sense in 1998 when a simple experiment was carried out for which Americans Andrew Fire and Craig Mello later won a Nobel Prize in Physiology or Medicine in 2006. These investigators injected double-stranded RNA molecules into the nematode. This resulted in the silencing of the gene whose sequence was complementary to the double-stranded RNA while silencing no other gene. The investigators called this very specific effect gene silencing, or RNA interference. So what's going on here? Well, a group of viruses called RNA viruses, those that contain RNA as their hereditary storage molecule rather than DNA, they replicate themselves through double-stranded intermediates. RNA interference may have evolved as cellular defense mechanisms against these viruses, which double-stranded viral RNAs have been targeted for destruction by RNA interference machinery. So without intending to do so, the nematode researchers had stumbled across this defense. Investigating interference, researchers noted that in the process of silencing a gene, plants produced short RNA molecules that matched the gene being silenced. Early researchers on far larger messenger RNA, transfer RNA, and ribosomal RNA had not noticed these smaller bits, tossing them out during experiments. These bits of RNA, called microRNA or miRNA, how do these small fragments of RNA silence the activity of specific genes? In a complex way, we are just beginning to understand clearly The small RNA fragments bind to any mRNA molecules in the cell have a complementary sequence. Silencing the gene that produces this mRNA is achieved in one of two ways. Either the expression of the mRNA is inhibited by blocking its translation into a protein, or the mRNA is destroyed. In either case, the specific gene that produced that mRNA fails to be expressed. It is silenced. So as you all continue on preparing for your test, please be sure to review putting the chapter to work and retracing the learning path as shown on page 224. Prepare for the test quite well, and this has been your instructor, Scholar Huff. I say thank you all for listening, and hopefully this has made much more sense.